Hello everyone, welcome to EPG Pathashala. I am Dr. Vinuta, scientist at Division of Biochemistry, Indian Agricultural Research Institute, that is IARI, New Delhi, India. Today I am going to talk about enzyme inhibitions. Under this module, we will be going to explore on enzyme inhibition, the nature of enzyme catalysis, and to understand in detail on various examples of enzyme catalysis. As you all know that enzymes are biochemically proteins and also RNA molecules. If it is RNA, it is called as ribozymes. These enzymes accelerate reactions and transfer energy forms without changing the reaction equilibrium. It catalyzes reactions by lowering the activation energy. What is activation energy? Activation energy is nothing but a minimum amount of energy needed in a system or from a reactants to initiate a biochemical reactions. Enzymes, biochemically proteins, accelerate reaction and transfer energy forms. Without changing the reaction equilibrium, it catalyzes reactions by lowering the activation energy. They are normally globular proteins and their tertiary structure creates an active site where substrate binds. The biocatalytic action depends based on the presence of some non-proteinaceous factors or cofactors. An enzyme is called apoenzyme if not bound by cofactor, and holoenzyme if it is bound by a cofactor. There are two basically different sorts of cofactors. Some are bound tightly, known as prosthetic groups, while some are entirely free of the enzyme and attach them to the active site alongside the substrate called coenzymes. The key concept of protein evolution has been the optimization of catalysis with versatile dynamics. Even though biochemical catalysis is similar to the chemical, they are more specific. It is carried out as an alternative reaction where enzyme lowers the energy barrier to reach the highest possible transition state. When the threshold drops, more reactant molecules achieve sufficient energy and get converted to products. In contrary to chemical reactions, enzyme is not consumed like substrate and then recycled for multiple rounds of catalysis. Let us see the features of enzyme catalysis. Specific base or acid catalysis. It means the chemical reaction catalyzed by acid or base. Acid is a proton donor or base is a proton acceptor. General acid or base catalysis. In this, any acid or base which participate in the mechanism of adding or removing a proton. The third one is neutralization of charge. Substrate get charged when bound to the enzyme and other residues of opposite charge clouds around the enzyme neutralize and helps to maintain binding. The fourth one is nucleophilic catalysis. Enzymes are always nucleophilic, either stronger or weaker nucleophile, or substrate acts as a electrophilic. This forms a covalent intermediate with the reactant. Other one is electrophilic catalysis. Withdrawing an electron from the reaction center results in electrophilic catalysis. ATP and metal ions are mostly perform electrophilic catalysis. Bond strain. The different types of bonds contribute to the enzyme catalyzed reactions. The strain is defined when a molecule is less stable due to electron repulsion or when not in ideal geometry. Different types of strains are transition strain, the steric strain and angle strain. Environmental effects. Any catalytic reaction can be accelerated or retarded due to the presence of solvents or other molecules surrounding them. Mechanism of alternative reaction routes. What are the different mechanism of alternative reaction routes? The first one is proximity and orientation. Catalysis will be performed when catalytic reduces in the active site come in close proximity to the chemical bonds in the substrate. 6 possible mechanisms of over the barrier catalysis are as well as thought the barrier mechanism have been explored till now. So, let us see what is proximity and orientation. Proximity or contact increases the probability of collision of two molecules. Orientation determines the position or geometry to react optimally. Both will increase the rate of reaction, allowing them to hold together and reduces entropy. Effect of proximity and orientation directly affect the randomness of the system and can be analyzed using computational studies. Let us see what are proton donors and acceptor in relation to enzyme catalysis. To stabilize the transition state and the charge associated, acids or bases accepts or donates protons. Among the amino acids, histidine normally involves with acid-base reaction and can therefore both accept and donate protons. So, in this table, it is given the effects of local environment on pKa. So, let us see what is electrostatic catalysis. The dissolution around active site decreases the dielectric constant and enhances the electrostatic interactions. Thus, the involvement of charged groups in maintaining the unstable intermediate favoring the electrostatic attractions is called electrostatic catalysis. Let us see what is covalent catalysis. In this mode of catalysis, substrate forms transient covalent bond with the catalytic residues or the cofactor. This covalent intermediate formed will reduce the energy of later transition state. Example, the catalytic amino acid residues of chymotrypsin and trypsin. These two, chymotrypsin and trypsin, are having an acyl enzyme intermediate. Preferential transition state binding or stabilization. Among the strategies, this is the most relevant in terms of enhancing the rate of catalytic mechanism. It is because enzyme tightly binds to the transition state than either substrate or product. Binding of enzyme to the substrate cause strain and distortion, which lead towards to transition state. The unfavorable interaction between substrate and enzyme is relieved by the formation of transient intermediate. Another modern concept of transition state stabilization states that it is not the substrate which gets distorted, but is the transient intermediate state, which favors better contacts and assists binding. The concept of induced fit assumes that the catalytic site of an enzyme is not complementary to the intermediate state in absence of ligand or substrate. Such enzymes will use some binding energy to support the conformational change and hence will have low turnover number. Let us see what is general acid-base catalysis. This includes general acid-base reactions like hydrolysis of peptide bonds, ester bonds and addition to carbonyl groups. Enzyme avoids an unstable transition intermediate by donating a proton or accepting a proton. Imidazole group in histidine, thiol group of cysteine and aromatic OH group of tyrosine and guanidine group of arginine, these acts as a functional groups with general acid-base catalytic nature. Let us see metal ion catalysis. Most commonly, it is an electrostatic catalysis or oxido-redox reactions. Metal ions activate bound water through the formation of nucleophilic hydroxide ion. Metal ion also acts as a Lewis acid or structural regulators. Example, carboxypeptidase A, aspartate transcarbamylase. Let us see the induced fit model. This model postulated a continuous change in the conformation of an enzyme after binding to the substrate. This promotes the formation of a tighter intermediate phase which lowers the activation energy and proceed to product at a faster rate. This explains the flexibility of active site and opposes the rigid lock and key concept. So now we will see what is quantum tunneling. This modified concept proposed in 1971 based on the observations that through the barrier as the first quantum mechanical model of enzyme catalysis, kinetics of certain enzymes operates so much faster than the predicted free energy change, mainly due to the tunneling of a proton or electron through activation barriers. Example is proton tunneling has been observed in tryptamine oxidation. Let us see some of the examples of enzyme catalysis. The first enzyme is acetylcholine esterase. An example for acid-base catalysis, this acetylcholine esterase inactivates the neurotransmitter acetylcholine by deesterifying. Serine amino acid is the sole responsible for cleaving the ester bond after getting deprotonated by a neighboring histidine. Another enzyme is carbonic anhydrase. This enzyme is a classic example of metal ion catalysis which catalyzes the carbon dioxide reacts with water to give bicarbonate and a protonated proton. This enzyme, known as the fastest in action, regulates the hydronium ion concentration in kidney during urine excretion or filtration. Zinc ions acts as the cofactor which helps to activate the bound water molecule to generate a hydroxide ion which can attack carbon dioxide. So another example of enzyme catalysis is serine proteases. These are also called as chymotrypsin. These are endoproteases that cleave peptide bonds, and the mechanism is simply nucleophilic attack by the oxygen radicals of water on carbonyl carbon of the peptide bond. The various steps in the mechanism of catalysis of chymotrypsin includes the formation of enzyme substrate complex. Enzyme binds to the substrate with specificity group R1 in a specificity pocket of the enzyme. This ensures the sterically appropriate position for cleavage of the peptide bond on carbonyl side of that residue. Formation of first tetrahedral intermediate. Aspartate in catalytic triad helps to maintain perfect steric binding with histidine and serine residues, and also facilitates H+ ion transfer by electrostatic stabilization of histidine H+ after it has accepted the proton. Formation of acyl enzyme intermediate. Histidine H+ ion donates a proton to the amino half of the substrate, and that amine product dissociates from the active site, that is P1 leaves the breaking of amide bond, results in the conversion of oxyanion back into carbon oxygen bond still covalently attached to the serine residues of the enzyme, forming the acyl enzyme intermediate. To summarize, we have learnt in this module the following points. Enzyme catalysis offers various features like specific base or acid catalysis, general acid or base catalysis, neutralization of charge, nucleophilic catalysis, electrophilic catalysis, bond strain and environmental effects. Mechanism of alternative reaction route are the various strategies to catalyze specific reactions. It includes proximity and orientation, proton donors and acceptors, electrostatic catalysis, covalent catalysis, preferential transition state or stabilization, general acid base catalysis, metal ion catalysis, induced fit and quantum tunneling. Various enzyme catalyzed reactions based on acid base catalysis. Metal ion catalysis, serine protease illustrate various mechanisms of action including proximity and orientation, transition state stabilization, covalent catalysis, electrostatic catalysis and general acid base catalysis. Thank you.